Good evening, one and all. This is Damodar. In previous lectures, we have discussed about bacteria and its history and the shapes, different shapes, and in detail structure of bacteria. Okay. Today in this lecture, we will learn about bacteria, nutrition, and types of bacteria basing on the staining technique. Okay, now let us see the type of bacteria basing on staining technique. Staining technique. So this staining technique of bacteria was first discovered by Christian Ingram. Okay, Christian Ingram named scientist first discovered this staining technique in a bacteria. So in this staining technique, what he has done? Okay, so in the staining technique, the stain he used is crystal violet. The stain he used to stain the bacteria in the staining technique is crystal violet. So how he has done the staining technique? So he has taken the bacteria culture. Okay, bacteria culture was taken by him. This bacteria culture is treated with our stain called crystal violet. Okay, so he treated this bacterial culture with the stain called as crystal violet. Now the bacteria that are present in this bacteria culture, okay, the bacteria that are present in this bacteria culture, so by taking this crystal stain, the bacteria turned into purple color. The bacteria turned into purple color. So here he stained the bacteria culture. That means the bacteria are present, so they are treated with the crystal violet. So this stain, by absorbing this stain, the bacteria turned into purple color. Next, this is the first step. Okay, this is the first step. In the second step, the bacteria turned into purple color, got fixed. Okay, got fixed. That process is called as fixation process. The bacteria that turned into purple color is undergone a fixation process with potassium iodide solution, with Ki solution. Okay, with Ki solution. So that what will happen? The stain crystal violet gets fixed to the bacteria. Now the bacteria which has turned into the purple color is treated with K solution. This is called as fixation process. As a result of this process, the stain crystal violet get fixed to the bacteria. That means bacteria completely the stain gets fixed onto this bacteria. Okay, so this is the second step. After this, in the third step, In the third step, after fixation process, bacteria are now treated with okay, the bacteria are now treated with acetone plus alcohol. Now in the third step, the bacteria which got fixed by this stain is now treated with acetone and alcohol. So when this is treated, when the bacteria gets treated with acetone and alcohol, okay, when here the bacteria gets treated with acetone and alcohol, now the bacteria shows two changes. Okay, the bacteria shows two changes. Okay, the one is some of the bacteria loses its purple color. Some of the bacteria loses its purple color, and some of the bacteria does not lose its purple color. Some of the bacteria loses their purple color, and some they does not lose the purple color. So the bacteria that loses the purple color are considered as gram negative, whereas that does not loses the purple color are considered as gram positive. Okay, so the bacteria now treated with acetone and alcohol. When acetone and alcohol is treated, the stain crystal violet is loosed. That means purple color is loose. So as a result, the bacteria which loses this purple color are considered as gram negative, and which does not lose the purple color are considered as gram positive, or called as gram positive bacteria. Now in the fourth step, after 1, 2, 3, now in the fourth step, what they will do is these bacteria are counter stained. These bacteria are counter stained with safranin. Okay, the bacteria, the gram positive and gram negative and gram positive bacteria are now counter stained with safranin. Okay, safranin solution. Okay, so now when safranin is, with the help of this safranin, it was counter stained. So the bacteria which is called as gram negative, okay, the gram negative bacteria turns to red. Okay, turns to red color, whereas gram positive bacteria does not loses its purple color. Okay, same, the purple color gets remained, whereas here the gram negative bacteria which loses its purple color now turns into red color. So those are called as gram negative bacteria and does not change. Still, after acetone and alcohol treatment, they won't lose the purple color. If they are again counter stained with saffron also, then also they won't lose this purple color. Such bacteria are called as gram positive bacteria. So these are the 4 steps. These are the 4 easy steps. So where staining technique of bacteria is done, which was discovered by Christian Ingram by using the stain crystal violet. Okay. So this is about the staining technique. So now by doing the staining technique, we identified two types of bacteria. One is gram positive and gram negative bacteria. Let us see the major differences of gram positive and gram negative bacteria. So now gram positive and gram negative. Okay, gram positive and gram negative. The major differences. So here cell wall of gram positive bacteria, cell wall of gram positive bacteria shows peptidoglycans of about 85%. Okay, 85% of gram positive cell wall shows peptidoglycans, whereas in gram negative shows only 5 to 10%, 5 to 10% of peptidoglycans, shows only 5 to 10% of peptidoglycans. Gram positive consists of 85% of peptidoglycans, whereas gram negative shows only 5 to 10% of peptidoglycans. Okay, so next, diamino pimlic acid is seen in some forms of gram positive bacteria. Diaminopimlic acid is seen in some forms of bacteria, whereas most of them shows diaminopimlic acid. Most of them shows diaminopimlic acid. Okay, next, third difference is teichoic acid. Teichoic acid is present in 10%. Okay, teichoic acid is present, which is of teichoic acid is present of 10%, whereas teichoic acid is absent. Teichoic acid is absent in gram negative. So this is a important bit in terms of competitive exams. Okay, so teichoic acid is present of 10%, teichoic acid is absent. Okay, next, lipopolysaccharides, phosphates and lipopolysaccharides, phosphates and lipids are present in minor quantity. Lipopolysaccharides, phosphates and lipids present in a minor quantity. Okay, so these lipopolysaccharides, phosphates and lipids are present near about 90 to 95%. Near about 90 to 95% lipopolysaccharides, phosphates and lipids are present in a gram-negative bacteria. So these are the major differences between gram-positive and gram-negative bacteria. Okay, these are the major differences. Cell wall consists of 85% of peptidoglycans, only 5 to 10% in gram-negative. Diaminopimnic acid is seen in some forms. Most of them shows diaminopimnic acid. Thyroic acid is present of 10%, whereas thyroic acid is absent in gram-negative. Lipopolysaccharides, phosphates and lipids are present in minor quantity, whereas consists 90 to 95% in gram-negative bacteria.